In this week's lecture, we are going to discuss another group of gram-negative rods. And unlike the previous gram-negative rods that we've discussed, this group of organisms are non-fermenting, whereas all the previous were fermenting gram-negative rods. This lecture is the last lecture that will be covered on your second exam, which is next week. And this lecture has been broken up into 3 different parts, and this is the first of the 3. So the clinically significant non-fermenting gram-negative rods, we have numerous organisms that fall into the Pseudomonas fluorescent group and the Pseudomonas non-fluorescent group. We also have several other organisms: Acinetobacter, Stenotrophomonas, Burkholderia, Flavobacterium, Cresiobacterium, Moraxella, Alligella, and Shewanella. We also have a whole slew of less commonly encountered non-fermenting gram-negative rods. Now, there are descriptions of almost all of these organisms in your textbook. However, I really think it's close to impossible to memorize every little tiny bit of minutia about every single possible organism that's ever been isolated from a human being. That it gets a little bit ridiculous. I know you're all already overwhelmed with the number of organisms that have to be covered in clinical microbiology. In my opinion, we are not going to go into detail in some of these less encountered gram-negative rods. You should be reading your textbook, so you will get some detail, but I'm not going to ask you very specific questions on these organisms. I think as long as you know which organisms are Enterobacteriaceae, which organisms are fermenting, which ones are non-fermenting, and then know the key features of some of the organisms that we discussed in more detail, that is enough. Now, I'm not going to tell you that there won't be a question on your board examinations on one of these organisms on this slide. There might be, but I personally don't think any one person can possibly know everything about every organism. I've been studying microbiology for a very long time. I'd say a good at least 15 years, at least, if not more, 20 years. And I don't know every tiny detail of every single organism. And I know a fair number of organisms. So I'm really just going off on this tangent just to let you know that this list right here, you should know some of these names and that they're non-fermenting gram-negative rods, but I don't expect you to know detail about these different organisms. So some of these less encountered gram-negative rods, you have Alcaligenes, Achromobacter, Chromobacterium, Comomonas, Flavimonas, Cresiomonas, Methylobacterium, Roseomonas, Ralstonia, Rhizobium, Sphingomonas, Sphingobacterium, Psychrobacter, and there's a bunch of CDC group organisms that haven't been specifically named yet. And I'm sure in the next year or so, more organisms will be named. If you actually look in your textbook, you're going to see that these organisms listed here, a lot of these were previously completely different organisms. So some of the names have changed around a bit over the last several years. Now, in general, here is a slide that you want to know. You really need to know this slide. These are the key characteristics of all of your non-fermenting gram-negative rods. Of course, they are non-fermenting gram-negative rods. That's something you need to know. The key thing about the non-fermenting organisms is There's a media we're going to talk about in this lecture that's called oxidative fermentative media, or OF media. These organisms will not acidify or turn the media to yellow in either the oxidative or fermentative tubes. Now, we've talked about some other tests when we talked about our enteric organism testing that will allow you to look at fermentation. So one of those is triple sugar iron agar. If you remember, the butt of that agar means if it acidifies, it means that the organism ferments glucose, and all of the Enterobacteriaceae members will be yellow in the butt of a TSIA. Any sucrose and or lactose fermenters will acidify the slant of TSIA. So if an organism ferments sucrose or lactose, there will be a yellow slant on TSIA. Now, these non-fermenting bacteria will not acidify a triple sugar iron agar tube in general. So in general, they're non-fermentative, so they will be alkaline over alkaline. They will not turn the media yellow. They will not be able to drop the pH and acidify due to the fermentation of the sugar. Now, we had other media that you could test fermentation with as well, like your McConkey agar allows you to detect lactose fermentation, and TCBS allows you to detect sucrose fermentation. We've talked about a lot of different medias that will allow you to detect various carbohydrate fermentations. The key media that you really need to know for the non-fermenters is this OF, or oxidative fermentative media, or the triple sugar iron agar, the key ones. Now, these non-fermentative gram-negative rods in general prefer to grow in an aerobic environment, and although they are non-fermentative, there are two different categories among the non-fermenters. Some of the non-fermenters are oxidizers, which means they can oxidize carbohydrates to derive energy for their metabolism. So they don't ferment carbohydrates like the Enterobacteriaceae or the Vibrio or Aeromonas do, but they oxidize carbohydrates. They can utilize carbohydrates in the presence of oxygen. Other non-fermenting gram-negative rods are non-oxidizers, or what's known as asaccharolytic or non-saccharolytic. This group of non-fermenting gram-negative rods do not break down carbohydrates or sugars at all. They are considered biochemically inactive. So none of these organisms ferment. Some of them oxidize, and some of them are completely biochemically inactive or asaccharolytic. Most of these organisms are oxidase positive, so that will allow you to differentiate these organisms from your Enterobacteriaceae gram-negative rod members, because all of those, remember, are oxidase negative. However, you need to be able to differentiate these organisms from your Vibrio, Aeromonas, and Plesiomonas, which are oxidase positive fermenting gram-negative rods. Now, these organisms, just like most of the gram-negative rods, are ubiquitous, meaning they're found in almost any environment. They are rarely part of the normal flora, but they can transiently colonize hospitalized patients, and these organisms tend to be resistant to various antimicrobial agents. Now, the oxidative and fermentative, and here's something that will allow you to understand the difference of the fermenting organisms from the non-fermenting organisms. So as you know from general microbiology, your bacteria will utilize glucose as well as other carbohydrates, sucrose, lactose, maltose, using certain metabolic pathways. Some bacteria use oxidative pathways, whereas others involve a fermentation reaction. So oxidative organisms can metabolize glucose or other carbohydrates under aerobic conditions where oxygen is the ultimate hydrogen acceptor. Fermentative organisms ferment glucose, and that hydrogen acceptor is another substance such as sulfur. So the thing about the fermentation process is it's independent of oxygen. So your fermentation can happen without any oxidation any oxygen at all, whereas oxidation requires oxygen. Now this media, and you really need to understand this media. It's going to be very important with your non-fermenting gram-negative rods, and in order to determine if something might be a fermentative or non-fermentative gram-negative rod. So this is our oxidative fermentative media, or OF media, and it is also sometimes called the Hohenliefsen test. So what this media is it's a semi-solid media that comes in tubes, and this media contains glucose as the carbohydrate, and it also contains a pH indicator, just like a lot of our differential media has in it. For each organism, you have to inoculate 2 tubes. That's imperative. One of those, you inoculate both tubes with your organism that you're testing. You take a needle, you have your semi-solid deep is what it's called, just the agar, semi-solid agar is sitting in the tube, and you use a sterile needle, you pick up your colony, and you just stab in the middle of the tube about 3/4 of the way down and come all the way back up, and keep that needle as straight as can be. Once you inoculate both tubes, one of those tubes you overlay with mineral oil. So you have sterile mineral oil in a dropper, and you drop a few drops so that you have a little layer completely covering that agar in the tube. You're then going to stick both tubes with a loose cap. You always want to have your caps loose when you incubate, into an incubator. So organisms that oxidize, they can utilize carbohydrates in the presence of oxygen, will produce an acid reaction in the open tube only. And the open tube means the tube that doesn't have the mineral oil over it. What the mineral oil does is it blocks oxygen from getting into the agar where the bacteria is. So you're generating an anaerobic environment with that mineral oil. So oxidizing organisms will produce an acid reaction in the open tube only, and there'll be no color change at all in the tube that has the mineral oil. Fermenting organisms, however, will produce an acid reaction in both tubes. So fermenters can utilize carbohydrates in the presence or absence of oxygen. So if an organism is a fermenter, like a Vibrio or like an E. coli, both the open OF tube and both the open media and the mineral overlay media will both turn color because you'll get an acid, lower the pH. Now, you're completely biochemically inert, or your non-saccharolytic or asaccharolytic organisms, they will not be able to change the pH in either the open or the closed or overlain with mineral oil tube. So here we're going to go through this again on this slide. So if you are utilizing your carbohydrates, you metabolizing those carbohydrates, the pH drops. When you produce acid, the pH indicator in the media turns from green, it starts out with green, and it turns yellow. So this tube, when you take it out of the box as a sterile tube, it is a green semi-solid agar inside of a tube. So you can see on the very far left, if you have an oxidative organism, meaning an organism that can only utilize carbohydrates in the presence of oxygen, you are going to get a yellow color change to the media in the tube that does not have the mineral oil, which is considered the open tube. The tube with the mineral oil stays green. Now in the middle picture, your fermentative organism. Fermenters can utilize carbohydrates in the presence or absence of oxygen. So fermenters are going to turn the media yellow due to the low pH in both the open tube and the closed tube. So the closed tube is the one again with the mineral oil. Then your non-saccharolytic organisms or your asaccharolytic organisms the media will stay green whether there's mineral oil or no mineral oil, so open or closed. So in the open tube, if you look at the chart in the middle, if you have an oxidative organism, the open tube will be acid or yellow. The closed or covered tube will be alkaline, which could be green or red. If the media is green, if the pH actually goes up instead of down, the green media can actually turn a red color. So if it's green or red, then that means there's no acid and no type of utilization. A fermentative, again, acid and acid, and the non-saccharolytic will be alkaline and alkaline, which both tubes will be green or green and red if the pH goes up. Now for clinical infections, the non-fermenting gram-negative rods account for about 15% of all of the gram-negative rods isolated from clinical specimens. They are responsible for a very wide range of infections, just like all of the gram-negative rods are. We've talked about all of these types of infections with the gram-negative rods. They can cause septicemia, meningitis, osteomyelitis, or bone infections, wound infections, especially following any type of trauma or surgical procedures. The common risk factors are the same with most of these organisms. Anyone who is immunosuppressed in any way, if someone has an implantation or a foreign substance in their body, like a heart valve or something like that, somebody that's had a trauma, someone that has a catheter or is getting fluids introduced to their body, things like that are always going to increase the risk of infection. Most of these types of infections occur in hospitalized patients or in recently hospitalized patients who have just been released from the hospital. Now, the biochemical characteristics of these organisms, they do not ferment carbohydrates. That doesn't mean they don't utilize carbohydrates. Remember, some of them are oxidizers, but they do not ferment carbohydrates. They will not yield an acid reaction in either the triple sugar iron agar or the Kliger's iron agar media. So we already talked about the triple sugar iron agar, and you can read up more on your Kliger's iron agar. They're somewhat similar types of reactions where you can look at hydrogen sulfide production and fermentation and things. So the most commonly identified or isolated non-fermenting gram-negative rods you will see in a clinical diagnostic microlaboratory will be Pseudomonas aeruginosa, Acinetobacter species, and Stenotrophomonas multifila. We're, of course, as usual, going to start discussing our most highly significant organisms. The pseudomonids are definitely the organisms that you would most commonly see out of all of the non-fermenting gram-negative rods. And we already said there are two groups that fall under the category pseudomonids. There's the Pseudomonas fluorescent group, which includes Pseudomonas aeruginosa, fluorescens, brutida, veronii, montilii, and mosellii, and the Pseudomonas non-fluorescent group, which is Pseudomonas stutzeri, mendocina, luteola, and rhizihabitans. Now, of course, we always start talking about the most notorious, the worst one of these organisms. And the worst or the most common cause of infection out of the pseudomonids is Pseudomonas aeruginosa. And actually, Pseudomonas aeruginosa is the organism that is going to cause the most human infections and the most common cause of hospitalized infections, hospital acquired or nosocomial infections out of all of the non-fermenting gram-negative rods. So Pseudomonas aeruginosa is highly associated with causing infection in burn victims. It can cause respiratory diseases such as pneumonia. Aspiration pneumonia is a very common nosocomial infection, and it also causes respiratory disease in cystic fibrosis patients. And cystic fibrosis is a genetic disease that causes a lot of different symptoms. One is an accumulation of mucus in the lungs, so these individuals are highly susceptible to respiratory tract infections, especially with organisms like Pseudomonas aeruginosa. It can cause ear infections or otitis media. It's a common cause of swimmer's ear. It can cause urinary tract infections, bone infections, and it is the third most common cause of bacteremia. So, Pseudomonas aeruginosa, you will see it in the clinical microlab a lot. There are certain organisms that are common causes of human infection that you're going to see in the clinical laboratory much more often than any other organism. So Pseudomonas aeruginosa you will see when you're working in the clinical lab. Now the pseudomonids are known to produce various pigments. So all of the organisms in the Pseudomonas fluorescent group produce a yellowish green pigment called pyoverdin. And, verdin, you're green. Pseudomonas aeruginosa, though, out of the fluorescence group, is the only organism that can produce a variety of other pigments in addition to pyoverdin. So Pseudomonas aeruginosa can produce pyoverdin like the others, but it can also produce pyocyanin, which is a blue pigment, pyorubrin, which is a red pigment, or pyomelanin, which is a brown to black pigment. So the identifying characteristics of Pseudomonas aeruginosa, is its ability to produce various types of pigments. Now, these pigments usually you won't see unless you plate the organism out on specific media. So in general, the pigments don't normally come out on typical 5% sheep blood agar. Usually you have to use other types of media like Mueller-Hinton agar. You can get some nice pigments coming up. The organism tends to be beta hemolytic on sheep blood agar that produces flat spreading colonies, and it does produce this very distinct metallic sheen. So if you take the blood agar plate and you hold it up to the light and you kind of move it back and forth while you're looking at it, you'll see this metallic sheen on the colonies. It's very distinct. Once you see Pseudomonas aeruginosa a couple of times, you will never miss it. You'll know it's Pseudomonas right away. Many Pseudomonas aeruginosa strains produce a fruity or a grape odor. Now again, there's no sniffing of plates in the clinical laboratory. However, when you have Pseudomonas aeruginosa and you open up that plate to pick a colony, you won't miss that grapey odor that it produces. So you will smell that grape-like odor. It will grow at up to 42 degrees Celsius. It is citrate positive. So remember we talked about the citrate test when we talked about the enteric testings. It's part of the IMVIC test. The Pseudomonas aeruginosa organisms are innately resistant to many antimicrobial agents, so they can be somewhat difficult to treat. And there is a selective and differential media called cetramide agar. So cetramide will inhibit most bacteria, but it allows Pseudomonas aeruginosa to grow, and it also enhances the pyoverdin and pyocyanin pigments. So you'll get nice green and blue colonies on this media. Now, some of our other organisms in the Pseudomonas fluorescens group, we have Pseudomonas fluorescens and Pseudomonas putida. These have low virulence. They are rare causes of clinical disease. They can be isolated from respiratory specimens, urine, as well as hospital equipment. They can grow at refrigeration temperature, 4 degrees, and Pseudomonas putida is gelatin hydrolysis negative, whereas Pseudomonas fluorescens is positive. Pseudomonas dezeri, another rare pathogen that produces wrinkled, leathery, very adherent colonies. The colonies adhere to the agar. They're very difficult to pick up off of an agar plate, and they can produce a light yellow or brown pigment. So Pseudomonas dezeri has been known to cause septicemia, pneumonia, endocarditis, conjunctivitis or eye infections, and urinary tract infections. Here's a gram stain of a bronchial specimen, and this is Pseudomonas aeruginosa. So you could see some of the mucus, you could see some degraded white blood cells, and you see a ton of gram-negative rods. Now, I've said this numerous times, and I'm going to say it again. You really cannot differentiate one gram-negative rod from another gram-negative rod using a microscope and a gram stain. So gram-negative rods are rarely gram stained. You'll know they're you'll have a gram-negative rod because it's growing on McConkey agar, not because you actually gram stain it. So in the clinical setting, some of the gram positives are gram stained because you can see variations and get an idea of what it might be. But in gram negatives, they all look very similar. So it's a waste of time to gram stain a gram negative rod usually. You will gram stain clinical specimens. So if you get a cerebrospinal fluid, you are going to gram stain it, and if you see gram-negative rods, you just call in and say, there's positive gram-negative, this spinal fluid is positive for gram-negative rods, so that the patient can be treated right away because they likely have a meningitis. But you will not be able to say, oh, this gram-negative rod on this scope looks like a Pseudomonas or looks like a Salmonella. You really can't differentiate like that from a gram stain. Here is Pseudomonas aeruginosa on sheep blood agar, and Pseudomonas aeruginosa can produce a non-mucoid colony type or a mucoid colony type. So the left is the non-mucoid, the right is the mucoid. Remember, the Pseudomonas aeruginosa is beta hemolytic generally. It discolors media, especially when it's in very heavily concentrated growth. Here's Pseudomonas aeruginosa on McConkey agar, and on McConkey agar you will start to see some of the blue and green pigments. And here's the pigments that you would see on other media like Mueller-Hinton or cetramide agar. On the left you have your pyoverdin, that yellowy green pigment, and on the right you have the pyocyanin or the blue pigment. Now another genus in the non-fermenting gram-negative rod is the genus Acinetobacter. This organism is a member of the Moraxaceae family. We've already talked about some of the members of the Moraxaceae family. Those like Moraxella cataralis. That organism is a gram-negative coccobacillus. So these organisms that we're talking about here are non-fermenting gram-negative rods. So that's why some of the Moraxella we're talking about in this lecture, even though we did talk about Moraxella cataralis previously. Now for Acinetobacter, there are two very common species. They are Acinetobacter baumannii and Acinetobacter loaffii. They are ubiquitous in soil, water, food, hospitals. They're pretty much everywhere. Adults commonly can carry Acinetobacter on their skin as well as in the pharynx. Hospitalized patients can be very easily colonized, so Acinetobacter tends to be an opportunistic organism. It will cause infection in hospitalized patients, people that are sick, people that are immunocompromised. So Acinetobacter causes 1 to 3% of all nosocomial infections. Not as many nosocomial infections as Pseudomonas aeruginosa, but still a fair number of nosocomial infections. And again, like Pseudomonas aeruginosa, it can cause urinary tract infections, respiratory, endocarditis, meningitis, conjunctivitis, septicemia. Now, Acinetobacter baumannii tends to cause more infections than loaffii. The identifying characteristics of the Acinetobacter species. These are oxidase negative, catalase positive, and they are small rods, coccobacilli to very short rods. They are non-motile. They're able to grow on most lab media, and some species have a purplish color on McConkey agar. They grow best at slightly lower temperature. So most of the pathogens, as you know, grow at body temperature, which is 35 to 37 degrees Celsius. These organisms grow best at 30 to 35 degrees Celsius. Now here's a key difference, and this is something you have to know. Notice I put different color text here. It means it's really, really important, and I can't put a neon sign in the slide, so this is as close as I can get. You really need to know this. Acinetobacter baumannii is saccharolytic, meaning that it can oxidize carbohydrates. It is a non-fermenter, but it can oxidize. Acinetobacter loafii is non-saccharolytic or asaccharolytic. It's one of those biochemically inert organisms. So that is a key test to differentiate these two species of Acinetobacter, so it's something you need to know. Acinetobacter loafii is more susceptible to antibiotics than Baumannii is. So Acinetobacter baumannii is saccharolytic. It does utilize carbohydrates, even though it doesn't ferment carbohydrates. So it can be turn pink on McConkey agar. So it can look like a lactose fermenter, even though it is a non-fermenting organism. It is oxidizing sucrose. Now, Stenotrophomonas multifila, this is the third most common non-fermenting gram-negative rod. So we have Pseudomonas aeruginosa, we have Acinetobacter, and we have Stenotrophomonas multifila. This is ubiquitous in the environment. It's very common in hospitals. It can contaminate blood drawing equipment. It can even contaminate disinfectants. So disinfectants that are used to wipe down surfaces to sterilize them, actually you can be putting a contaminant onto your surfaces if your disinfectant is contaminated with something like Stenotrophomonas. This organism is not part of the normal flora, but it can colonize the respiratory tract of hospitalized individuals. It's resistant to cephalosporins, penicillins, carbapenems, and aminoglycosides, and it causes the same variety of diseases that Pseudomonas and Acinetobacter can cause. It can cause wound infections, bacteremia. Rarely, but it has caused urinary tract infections, meningitis. It's been involved in causing endocarditis, especially in individuals who've recently had a heart surgery or in injection drug users. And it is isolated from 6.4 to 10% of cystic fibrosis patients. So Pseudomonas aeruginosa is one of the most predominant organisms that's isolated from cystic fibrosis patients, but Stenotrophomonas is another organism that causes infection in cystic fibrosis patients. So how do you differentiate your Stenotrophomonas? It is oxidase negative. That's a little bit different. So Stenotrophomonas can be tricky. Stenotrophomonas is oxidase negative. Well, your Enterobacteriaceae are also oxidase negative. So you've got to be very careful with not immediately. I mean, usually if you see oxidase negative, gram negative rod, you really do want to think Enterobacteriaceae. But if Stenotrophomonas is one of the answers in the mix, you always have to think about some of the other test reactions in addition to that negative oxidase. So Stenotrophomonas is positive for catalase, esculin, DNase, and gelatin hydrolysis, and it is susceptible to the SXT antimicrobial disc. So susceptible meaning there will be a zone of inhibition. So we're going to move on to the second part of the non-fermenting gram-negative rods.